Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 190 for the podcast. Today we welcome Dr. Rosanne Dunn, Chief Scientific Officer and co-founder of HaemaLogiX, an Australian clinical stage biotech company that's developing next-generation immunotherapies for multiple myeloma and other plasma cell disorders. HaemaLogiX is taking a precision approach by targeting novel antigens that are expressed exclusively on malignant plasma cells, sparing the healthy ones. Recent peer-reviewed research has validated KMA and LMA as high-value targets, reinforcing the company's unique positioning in the field. Rosanne shares her journey from antibody engineering to building HaemaLogiX, the science behind their differentiated targets, the latest clinical and preclinical progress, and her perspectives on the future of immunotherapy for blood cancers as the company gears up for key milestones, including a planned IPO later this year. It's an exciting time for Australian biotech innovation on the global stage. Let's dive in. Rosanne, welcome to Beyond Biotech.
Thanks, Dylan. Very nice to be here.
You know, it's always fun for me as an Australian to get back and talk to some Aussie biotechs, especially ones that are starting to make a global impact. Maybe, though, we can start with you and your career journey. What first sparked your interest in science?
Right. So I actually did my undergraduate degrees. Firstly, my honours degree in biochemistry at the University of Cape Town. And then I did a Master's of Medical Science at the University of Natal Medical School. And then subsequently, I moved to Australia and did my PhD here at the University of Technology, Sydney. Now, during my undergraduate years, I was introduced to molecular biology and recombinant DNA technology. And I found these topics fascinating. You know, in particular, the opportunity to use these technologies for clinical purposes, the identity and, and understanding of diseases that have a genetic basis, and also being able to modify proteins that you could actually create therapeutics with. So that essentially was why and how my interest was sparked in science.
Mm-hmm. And you co-founded HaemaLogiX. You've been a director right from the start. What motivated you to actually start a company, though, and dive into the business side instead of just the academic, the research side?
Sure. So in the mid-90s, I was fortunate to be offered a PhD project that involved recombinant DNA technology and the creation of a recombinant immunotoxin from the original antibody that targets Kappa myeloma antigen and has subsequently been humanized to produce Kappa map. Now, back in that era, sort of the late 90s, early 2000s, it was a really exciting era for, for antibody engineering, in particular the humanization of monoclonal antibodies using CDR grafting, for the treatment of cancer and autoimmune diseases, for example, rituximab. Also the, phage display, which is the creation of novel human antibodies that didn't require hybridoma technology. And this was an example is Humira, the anti-TNF alpha antibody. And then, of course, the transgenic mouse platforms that created fully human monoclonal antibodies, such as ipilimumab and the anti-PD-1 checkpoint inhibitor monoclonal antibodies, such as nivolumab. So the reason why, I sort of decided that I was motivated to actually start a company was seeing all of these therapeutics and immunotherapeutics actually coming into the market and into the clinic, well, mostly into the clinic, but also into the market to treat, these diseases, and seeing the relatively safe profile of these monoclonal antibodies in the treatment of cancer and autoimmune diseases. So that was really, it was that era of, immunotherapies coming into the field, if you like, that sort of sparked that idea of starting a company. But also, the other reason was that, and this is serendipity, I mean, this is the backstory, right, is that we were doing an in vivo mouse model with the monoclonal antibody as a control, and the immunotoxin that a colleague of mine had created in a mouse model for myeloma. And it was the control antibody, the native antibody, that was very effective at preventing the tumor from growing. So once again, this was something that sort of sparked the interest of creating a company, spinning it out. And in fact, that was, the topic of our very first patent was using capimab as a therapeutic for multiple myeloma.
You were involved in discovering that lead therapeutic antibody now, that capimab, taking it from the lab all the way into the clinic. I mean, that's got to be pretty rewarding.
Yes, it has. It's been very rewarding and like really interesting all along the journey. So we started off, as I say, with, a mouse hybridoma or a monoclonal antibody from a mouse hybridoma, and then we humanized that. We then, had an agreement with a company called Medarex that actually made the monoclonal antibody for our first phase 1 and 2 clinical trials. And, then subsequently, we have actually now taken that antibody through to a phase 2b clinical trial that we've recently completed. So all along the way, we've had, we've had a really, if you like, interesting journey where things have worked, well in that we've shown that the antibody is safe, it's well tolerated, it is efficacious in combination with a standard of care drug called Revlimid. But most importantly, we've actually also had the support of a number of key opinion leaders and hematologists as we've moved through from preclinical and into clinical development.
I may have skipped forward a little bit there. So for listeners who are new to the company, maybe explain a little bit of what HaemaLogiX is and what the mission of the company is.
Yeah, sure. So, HaemaLogiX is a biotech company based in Australia. And essentially what the mission is to develop our immunotherapies. First of all, as I say, we've got our core focus, which is the antibody against the kappa myeloma antigen. And essentially what we've, let me step back a bit. HaemaLogiX, we've developed these or identified these novel myeloma antigens that appear as two variants. So there's a kappa myeloma antigen and there's a lambda myeloma antigen. And just as the name suggests, these are antigens that are specific to the clonal plasma cell that expresses a kappa light chain and a lambda clonal cell that expresses a lambda light chain. But interestingly, what we've demonstrated in the lab and also confirmed with our clinical trials is that capimab is very specific for KMA. And in the clinical development, we've shown that, in patients whose disease has relapsed and is progressing, this specificity has been demonstrated, whereby the antibody is actually efficacious and can prevent the cancer from progressing, the myeloma from progressing. But not only that, we haven't observed and we haven't seen evidence of hematological toxicities and or lymphodepletion. So I think we've this is our aim, if you like, is to develop these immunotherapies against these novel antigens that we've actually identified. Multiple
myeloma is really the second most common blood cancer. There's obviously still no cure there. Can you tell me a little bit about the disease? What does it do? How does it surface?
Sure. So multiple myeloma is actually a blood cancer, but it's a cancer of the plasma cells that proliferate in the bone marrow. And essentially, as they proliferate, they cause a number of symptoms, if you like. For example, there's hypercalcemia, because what happens is the myeloma cells actually interfere with the osteoblasts that lay down the bone. So you get actual, lesions within the bone. So that causes this hypercalcemia. There's also renal involvement because of the M protein, the free light chain that actually causes depositions within the kidneys. And so you get renal involvement. And then, of course, there's anemia, which again that's as a result of the plasma cells actually almost overcrowding the bone marrow and preventing the normal hematopoiesis. And of course the bone lesions, which as I mentioned, that's to do with the hypercalcemia as well. So the bone lesions actually can be, in some cases, fatal, because essentially it weakens the bones, and actually you can have really severe fractures. So that's essentially what it is. But most importantly, I think, is the proliferation of the plasma cells in the bone marrow, because most of the therapeutics do is actually try to deplete those plasma cells that are growing in the bone marrow, or to stop them from growing. And because of that, some of these treatments can actually be quite toxic. And we can go into those sorts of, issues as we move through.
Yeah, I guess one of the things that's special about your approach is it's targeting only these malignant plasma cells. It spares the normal ones. Is this something that sets HaemaLogiX apart from other therapies, other approaches?
Yeah, it certainly is, because what we've shown is that, these targets are not on normal plasma cells. And that means that the normal plasma cells are not depleted with this treatment. And the most important role of a plasma cell is to make antibodies against any sort of, pathogen, if you like, a bacteria or a virus, or, sometimes even fungus. So those antibodies are crucial in defending the body against those sorts of infections. Now, if you deplete all of those normal plasma cells, the ones that are there to make those antibodies against the diseases, then of course, the patients are likely to become, susceptible to life-threatening infections, because you've actually depleted their normal immunity against these things. So one of the issues that some of these, certainly most of these therapeutics against the myeloma cells, one of the side effects is that they suffer from infections, right? And eventually they, deplete, obviously, the immunoglobulins that the antibodies make, which is part of the immune, surveillance against those sorts of bacteria and viruses. And one of the issues can be that, patients actually die from the infections and not from the progression of the myeloma disease. So that is something that we haven't observed as yet, or in any way with Capimab, and we don't expect to, because of the specificity of our therapeutics.
Can you give me a description of what the pipeline at HaemaLogiX looks like right now?
Yes, certainly. So as I mentioned, we've got Capimab, which is a monoclonal antibody that is targeting our kappa myeloma antigen. And that's in a phase 2b study that is going in, we're going to initiate that in the next, this year, early this year. And that is going to be in combination with another standard of care drug called pomalidomide and dexamethasone, both of which are approved for this patient population where, the patients will have failed 3 classes of drug, including an anti-CD38 antibody, an immunomodulatory drug, and also a proteasome inhibitor. So that is a combination that we're actually about to use with a higher dose of capimab. The interesting thing is that pomalidomide is actually more effective in its synergistic effects with capimab because it actually increases KMA more on the surface of the myeloma cells more than Revlimid does. So potentially it could be a better combination or more synergistic for these patients with a higher dose of capimab. So that's where we are with that. It's our phase 2B that we're about to initiate. And then we've got a KM CAR T cell. So that's using our monoclonal antibody as a single chain FV to target a CAR T cell, so the chimeric antigen receptor. And that's in a T cell, it's an autologous treatment, the patient's own T cells. And that we've had some really excellent preclinical data showing that the KM CAR T cell retains the specificity, it's efficacious, and it actually is, it does actually prevent the tumor from growing over time in an in vivo model. So that is going into a phase 1 clinical trial at the Peter MacCallum Cancer Centre in Melbourne. And that, importantly, is a collaboration that we have where the Peter MacCallum actually has a government grant that they will be using towards the funding of that CAR T cell. And it's a dose escalation study. It's a typical phase 1 dose escalation study where, we increase the dose over a number of cohorts of patients and then find the efficacious but also tolerable dose of the CAR T cell and expand in a total of 12 patients in the study. So that is that's our pipeline for our KMA targeted therapeutics. And then, of course, we have the lambda versions of our immunotherapy. So we've got lambda monoclonal antibodies that we've now been through several preclinical studies with. These were created against the target LMA. And we know that they're effective against myeloma cells and also against amyloidosis or plasma cells from patients who've got amyloidosis. So we know that, from a preclinical perspective, these lambda mAbs are in development, if you like, for early studies. And then we do have some early data on our LMA CAR T cell or LM CAR T cell, which again is using the single chain FVs from these very specific monoclonal antibodies that we've developed to target, LMA in myeloma, but also in amyloidosis. And we've also, from a discovery perspective, we've developed bispecific antibody using the capimab version of the antibody. And of course, we will move forward on the lambdas as we prove, from a preclinical perspective, prove it in the lab as well.
It certainly sounds like a robust pipeline. One of the reasons I wanted to have you on is because of some research that just got published. You just announced some peer-reviewed research published in the journal Clinical Lymphoma, Myeloma, and Leukemia. In really simple terms, what did the study set out to investigate?
Right. So we've actually proven with capimab, we've shown KMA, from a preclinical, clinical perspective, and etc. But the lambda mAbs are much earlier in development. So from if you like, a proof of concept perspective, what we wanted to show that there was, the KMA, the target kappa myeloma antigen, the lambda myeloma antigen actually mirrors that, except it's a lambda version. And the idea was to look at a large cohort of patients. It was 195 patients, but not only look at patients who had myeloma, who had active myeloma, but there's a pre-malignant phase in myeloma, which is called monoclonal gammopathy of undetermined significance. So essentially, those patients actually have the potential to develop myeloma. There's also a type of as myeloma progresses from MGUS, we call it, right, to smoldering myeloma, where the patients in that group are more likely to develop myeloma than the sort of more indolent MGUS patients. And then, of course, there's newly diagnosed, and then there's patients who've been treated. So we wanted to look across that spectrum from early diagnosis, very early diagnosis of an abnormal plasma cell, all the way through to what a patient who's been treated plasma cell looks like, and what happens to these targets across that spectrum. And we showed that they actually they are expressed, both KMA and LMA, in that early stage of MGUS all the way through to relapsed refractory. And in fact, in the relapsed refractory patients, or in the patients who've been treated, let's just say that, the density of these targets is higher than that of BCMA, which is a target that is currently used for both CAR T cells and bispecifics to treat myeloma. And we also wanted to look at a number of different types of the plasma cell, they call plasma cell dyscrasia. So these are abnormal plasma cells, one of which was light chain amyloidosis. And again, we showed that, in fact, in particular, the LMA, because step back a bit here, in amyloidosis, the majority, 70% of them, are lambda light chain isotype, right? And we showed that, in fact, LMA is much more highly expressed on the plasma cells from amyloidosis patients compared to BCMA. And in some cases, the plasma cells didn't even express BCMA. So, and likewise with some of the myeloma patients, KMA was expressed and BCMA was not expressed. So this is why we really wanted to have a look at where are these targets expressed and compare them to the current or the targets that are currently used for immunotherapies. And we also showed that there was, if we looked longitudinally, so imagine a patient who is initially diagnosed with monoclonal gammopathy, then progresses through to smoldering myeloma, and then progresses through to, active myeloma over a period of 2 to 3 years. We had two patients who had that sort of longitudinal development. And we showed that as the patient progressed from, MGUS to smoldering, the number of LMA targets actually increased dramatically, more so than BCMA. And certainly by the time they had overt myeloma, the number of targets was, LMA targets was in the 90% sort of thing. So it's the density of the target on the plasma cell is much higher than BCMA, but also they are important in the disease progression. Those cells appear to expand more over time.
If this research is showing that, these antigens are present right from the really earliest pre-malignant stages, the MGUS stages as well, and they increase as the disease progresses, what does that mean for when treatment might actually start? Is there a chance we can start treatment even earlier than we do now?
Yes, very much so. One of the reasons that clinicians don't treat patients with MGUS is because they don't necessarily progress to myeloma. So if you have a toxic drug, there's no reason to give a patient who's not going to end up with active myeloma a toxic drug. So having these immunotherapies that have really specific or are highly specific, but also are have a really good safety profile and are well tolerated as far as side effects are concerned, that has the potential for us to move closer into early treatment, first line treatment, or even in patients who are sort of potentially likely to progress to smoldering and then on to myeloma.
And if it's also true that the standard therapies, the Revlimids, they can already boost the expression of KMA and LMA, does that mean in combination you could have a therapy here that could improve outcomes in the end?
Absolutely. And one of the interesting things about, the treatment of myeloma is that about 40% of patients will have an autologous stem cell transplant as their first line of treatment. And Revlimid is used as a maintenance drug. So after autologous stem cell transplant, Revlimid is used as a maintenance drug to prevent the myeloma from recurring. Now, it's not, certainly it does actually prevent the cancer from coming back, but it certainly isn't a cure post autologous stem cell transplant. So this would be an ideal position for our monoclonal antibody because the standard of care is Revlimid. We know that it is synergistic with our antibody, and our antibody we know does not add any toxicity onto the Revlimid backbone.
It really seems to validate what you're doing. I mean, this must have been a great paper to be able to pull together and validate the approach and validate the pipeline there. Does this mean then that you're moving into this phase 2b study with a whole lot more confidence? Is there something specific you're really hoping to get out of the phase 2b to set yourself up for the next phase of clinical trials?
Well, yeah, we are moving into the phase 2b with more confidence because we certainly shown without a doubt that our antibody is highly specific for this target, and as I said, we're going to increase the dose because we've shown that, in fact, the dose we were using previously was really well tolerated, and we're increasing the dose because we're using pomalidomide, which again is going to increase the target KMA more than Revlimid. So we've got a lot more targets. We need a lot more. Not a lot more, but we need to increase the dose of the drug. It's really safe, and potentially this could be a move into, if it's successful, right? This phase 2b, it could potentially move into a pivotal phase 3 clinical trial because these patients, they've failed multiple classes of drug, and there's a clear unmet medical need, and potentially this could be a pivotal clinical trial for our monoclonal antibody.
And the phase 2b is all in Australia, or are you going overseas as well?
Initially, it's in Australia, but of course, we are raising the money to move forward and to, hopefully take this into a global study. So
yeah, it sounds fantastic. I mean, what's the impact on patients for therapies like this? What's it going to do to their quality of life? What's it going to do to addressing the myeloma?
Well, I think the most important thing, obviously, is to be efficacious, right? And I mean, we do have anecdotal evidence that a couple of patients post the previous clinical trial stayed on our antibody drug and were disease-free for up to 4 to 5 years. We do know, obviously, working with our, principal investigator, Professor Andrew Spencer, that a particular patient we still provide the drug, we still provide Capimab. It is coming up to 6 years disease-free with absolutely no side effects related to the monoclonal antibody, but obviously with Revlimid, dexamethasone, there is some mild toxicity, neutropenias, that sort of thing. And, the patient's quality of life, hopefully, we're going to improve all of their lives in that way, That's really important.
And I guess for the patients at those really early stages, the MGUS stages, that gives them a little bit of certainty in a period where they might otherwise be told, well, we're just going to watch it, we're going to wait and see what happens.
Absolutely. Because we can, just by doing bone marrow aspirates, we could actually see, is that population of KMA positive cells, is that growing? And then that potentially you want to treat that patient. But, just going back to the quality of life, I mean, sometimes we don't realize that patients who don't really have, who are immunocompromised, so they don't have a robust immune system, they really are, limited in where they can go out. I mean, for example, some of these patients are elderly, and just, if your grandchild or your child has a viral infection or a pneumonia or something like that, or there's someone in the community, I mean, you're not really, it's not going to benefit you going out and getting that disease because you really don't have an immune system to cope with that. So from a quality of life perspective, I think that's the important thing, is being able to mount a response against an infection, and being able to live a normal life out in the community.
You mentioned a couple of minutes ago that looking forward, this was so important for the company. What are the major milestones that you're aiming to hit in the next couple of years?
Sure. So the major milestones obviously are the development of our monoclonal antibodies, in particular for multiple myeloma, with the Capimab and the Lambdamab, and also the CAR T cells, right? I mean, those I think cell therapies have a very very important role to play in the future. I mean, development of treatment for these diseases, in particular hematological diseases. And there's a multitude of different platforms that CAR T cells are actually now moving forward. Different ways of targeting the same target. I don't know if you've seen sort of like Arcellx was bought by Gilead, I think it was, and that's using D domains, right? Which is a form of, it's really just a targeted, it's a platform, a protein platform that targets BCMA and potentially, is better than the single chain FVs of BCMA, etc., or the camelid antibodies. But not only that, there's these new platforms, in vivo CARs, which potentially, it takes a step, it sort of leaps over the manufacturing step that you have to do with autologous T cells, CAR T cells, and you simply, either provide the a nanotechnology mRNA or actually a lentivirus that you can actually direct it to patients' T cells themselves. So there's no manufacturing of the CAR T cell or CAR NK cell or whatever. So I think cell therapies is a very interesting platform, and we want to continue to explore the CAR T cell platforms for both the antibodies, the Capimab and Lambdamabs. And then, of course, the bispecifics. Everyone thinks that, potentially, because they're in a, like monoclonal antibodies, they come in a vial, and therefore, you can use them, and then you could stop if it's toxic to the patients or whatever. So we are continuing to explore our pipeline, to explore novel platforms, and these are important for us going forward.
That's all on the clinical side, but on the business side, you're also planning an IPO in 2026. How is this going to accelerate the development and push you towards meeting those goals?
Right. So it's very important, in particular for the monoclonal antibody, the phase 2b clinical trial. I mean, that is the reason why we are raising the money, is to potentially initiate that phase 2b clinical trial and move forward into, given success, right, move forward to, a sort of a pivotal clinical trial proving that this combination that we've designed is going to be effective in this patient population. So that's the majority of the funding will be for that. And then, of course, as I mentioned, the KM CAR T cell, now that is currently being funded. The phase 1 clinical trial is being funded at the Peter MacCallum Cancer Centre through a federal government grant. And once we've reached 12 patients, clearly we need to, move forward, if you like, for, phase 2 studies. The other really important development program that we have is the lambda maps. For amyloidosis, this would be ideal. And we have done some research with Professor Jesus and Miguel that it was published as an abstract showing that the lambda maps are very effective in, destroying the malignant plasma cells of patients, amyloidosis patients taken from their bone marrows. So, there's a lot that we can do, and certainly we need the money to do that. So that's the point of our IPO.
From your perspective, Rosanne, how do you see the field of immunotherapy for these blood cancers evolving over the decades to come?
Sure. So, I really think that, we need new targets, and we need specific targets for immunotherapies. And this is why, it's really important for us to continue with the development of our program, because even if the platforms, for example, if the monoclonal antibodies are, translated into bispecifics, and you can actually stimulate the immune system with a T cell engager or an NK engager or something like that, you still need a relevant target, something that's not going to actually cause, potential harm to the patient, but also that's not eventually going to be, it's going to be effective, because some of these under selection pressure in particular has been shown that BCMA actually is deleted from the chromosomes, and you have mutations, et cetera. So I think we desperately need new targets, and some of the targets, or they all come with baggage, right? They all come with side effects that essentially then patients need to be treated with something to overcome those side effects, whether it's intravenous immunoglobulin or sort of constantly on antibiotics, etc. So I think we need new targets. But I think there's a lot of interesting platforms in cell therapies coming through. As I mentioned, the in vivo CAR T cells, potentially those could be game changers. Bispecific and trispecific antibodies. I mean, we're also looking at trispecific antibodies because, with our targets, we could actually combine with these other targets that are not quite as or toxicity free, if you like. But we could always, it's a case of if it works better together, then absolutely, then we would, a trispecific or dual targeting CARs, So I think there's a lot that can be done with the current platforms and with, sort of, if you like, validated targets, but also with our novel targets.
What about these Australian biotech companies like HaemaLogiX? Can you really play a role on the global stage from somewhere that just seems so far away from Europe, from North America?
Yeah, that's one of those things, isn't it? Um, from down here, yes. Well, yes, of course. I mean, if you look at, the Australian biotech industry, there's a lot of innovation that happens in Australia. Uh, I think, the academic institutes actually really encourage innovation. Certainly, that was my experience. So there's a lot of really good science happening here. There's a lot of support from the academic institutions. And then, of course, there's a lot of government support as well for early preclinical and clinical development, because we have the R&D tax incentive, which really, if you spend money on research and development for clinical programs, then you get a substantial amount of your dollar returned. I think it's about 40% of that is returned to you through the government R&D tax incentive. So certainly Australia encourages innovation. There are a lot of good ideas here, and I think the what we really need is from sort of that early stage development where there's a lot going on. There's a lot of really good companies, a lot of good technology. It's that step to get to a really if you like, the pivotal stage. That is where certainly I think we need to be more global. But that's I think hopefully going to come eventually.
Finally, Rosanne, where can people go to learn a little bit more about the work that HaemaLogiX is doing?
On our website, HaemaLogiX.com. And of course, we are on LinkedIn. HaemaLogiX has a LinkedIn account. So we are actually, announcing quite a few things on LinkedIn at the moment. So it would be great if, people wanted to understand more about us and what we're doing at the moment. LinkedIn's a good place to start. And then YouTube as well. We've got quite a few videos on YouTube. But certainly our website gives a lot of information. And of course they're always welcome to contact us through info at HaemaLogiX.com.
That email was info@HaemaLogiX.com. Perfect. Rosanne, it's been a pleasure to talk to you this morning. Thanks for being on Beyond Biotech.
Thanks so much, Dylan. It was lovely to speak to you.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.